In this lecture, we're going to be talking about blood and the components of blood, particularly those that are responsible for blood clotting as well as the immune system. So first of all, if you take a blood sample, as we have here, and put it in a centrifuge and spin it down, this is something that's discussed in your lab. What you find is that the red blood cells, of course, because they are solid and denser than the fluid component, will settle to the bottom of the tube. And on top of that is going to be primarily this column of fluid, which is plasma. Plasma is about 55% of the blood volume. So the total height of this column of fluid, about 55%, is plasma. Plasma consists of water and proteins, and we'll be talking about the proteins that are in plasma in a few slides. The remainder, that is about 45% of this blood volume, is erythrocytes. So that is what we show here with our red portion of the column, representing red blood cells, which of course are important for transportation of oxygen. We can determine how many erythrocytes a person has by measuring what we call the hematocrit. The hematocrit basically represents the percentage of the blood volume that is represented by red blood cells. And the textbook value is 45%. What we find is that in men it ranges between 42 to 52%, and in women it ranges between 37 and 47%. Now, of course, these two values, 45 and 55, add up to 100. These are rounded up. There is a small area here shown called the buffy coat, and I refer to this again in your lab. This buffy coat is composed of platelets as well as leukocytes. So let's talk about them. Platelets are actually not cells. They are fragments of what's called a megakaryocyte, which is a very large blood cell. And they are involved in coagulation or blood clotting. We'll be talking about that in a little bit. And then leukocytes, which are part of the immune system. These are the white blood cells. Leukocytes are subdivided into several cell types. We'll talk about these later on. The eosinophils, the basophils, neutrophils, monocytes, and lymphocytes. Excuse me. Now, we've discussed body fluid compartments back at the beginning of the semester, but because it's pertinent to our discussion of plasma, we'll go over it again. So if you take all of the water that exists in the body, this is what we call the total body water, about 66% of this is intracellular fluid. And intracellular fluid is, of course, as the name suggests, the fluid that resides in each and every cell throughout the body, not only in organs and tissues, but even the fluid that's in the red blood cells themselves. The remaining third of total body water is extracellular fluid. Extracellular fluid consists of both plasma and interstitial fluid. The interstitial fluid, which is a larger proportion of the extracellular fluid than is the plasma. Interstitial fluid is the fluid that exists in the spaces between cells. These would be what we call the interstices, that is, spaces between cells. That's where the name interstitial fluid comes from. As well as all of the fluid that exists in any body cavity. The plasma, which is the smallest proportion of the extracellular fluid as well as total body water, is the fluid, that's the pink fluid here, that the red blood cells are suspended in. Of course, meaning that it is the fluid that exists here in our column above the red blood cells. Now, this is important because as cells consume oxygen, that oxygen, of course, is taken from the interstitial fluid, but it is replenished by taking it from the plasma. The cells, of course, produce CO2, so CO2 builds up in the cells, it moves into the interstitial fluid, and then moves into the plasma. Glucose being consumed by cells. Cells consuming it here is being replaced by glucose from the the interstitial fluid, which is, of course, being replaced by glucose that was in the plasma. So what I'm emphasizing here is the fact that these different body fluid compartments that are shown here are in constant interplay, that substances are constantly moving between one compartment and another compartment, and in fact, another compartment. So that it is important to realize that suppose you have a patient that's become dehydrated, so their cells have reduced the amount of intracellular fluid. If you give them fluid, let's say you give them an intravenous fluid, the fluid, of course, first of all, is in the plasma. It moves from the plasma into the interstitial fluid. Now, we've talked about this before in terms of microcirculation and fluid movement between the capillaries and the tissue. So we have movement of fluid from the plasma into the interstitial fluid, but then it's moving from there into the intracellular fluid. So that as the cells absorb more water to bring them back up into a non-dehydrated state, of course, we're taking away that moisture from the interstitial space and taking it from the plasma. So there's a constant movement back and forth of materials among these three fluid compartments, and that's something you need to keep in mind. So again, talking about plasma, 90% of plasma is water, and only about 6 to 8% of it is proteins. The proteins, and we'll talk about them in a bit more detail in a few more slides, but they consist of albumins, globulins, and fibrinogen, excuse me, as well as electrolytes. Plasma is relatively high concentration of sodium and chloride, and relatively low concentrations of hydrogen ions or protons, bicarbonate, potassium, and calcium ions. And of course, when we talked about movement of materials back and forth between the body fluid compartments, these electrolytes also move back and forth depending upon their concentration gradients and changes that may exist in the utilization of these electrolytes or the utilization of water in these different body fluid compartments. And for those of you that are going into clinical healthcare professions where you'll be dealing with patients that may have dehydration, etc., you need to realize that this is a very dynamic system and that materials move back and forth between these compartments, that these concentrations are not necessarily constant, that depending upon utilization or production of different ions, that these concentrations can change because of movement among these different body fluid compartments. So let's talk about the functions of plasma proteins. Generally, they contribute to the colloid osmotic pressure. We talked about these, or this, when we talked about the Starling forces that are involved in exchange of material in the microcirculation, that is between the blood and the tissue surrounding the blood. Also, plasma proteins buffer hydrogen ions, so they help to control pH. They increase the viscosity of the blood, and they can be used as a source of fuel or energy during states of starvation. Specific functions for the albumins, they are a major contributor of the plasma oncotic pressure that we talked about over here, the colloid osmotic pressure, which is the same thing as oncotic pressure. And they act as carriers of signal molecules, especially lipophobic molecules such as hormones. There are the globulins, which consist of two major groups, the alpha and beta, as well as the gamma globulins. Alpha and beta globulins also act as carriers of signal molecules. They contribute to blood clotting. We'll talk about that in a few more slides. Some of them act as enzymes, and some of them are precursor proteins, such as angiotensinogen, which we'll be talking about in considerable detail when we get into the renal system, which is involved in blood pressure control. Gamma globulins, another name for them is immunoglobulins. Another name yet for them is antibodies. We'll be talking about them in a little bit of detail before the lecture is over. These are, of course, part of the immune system. And then the last major group of plasma proteins is fibrinogen, which is involved in blood clotting. And again, we'll be talking about that in some more detail in a few moments. So erythrocytes, of course, red blood cells. 1 milliliter of blood, or 1 cc of blood, has about 5 billion. So we have many, many erythrocytes. They have a biconcave disc shape that is rather than being just a simple hockey puck sort of shaped structure, that in fact the two surfaces curve inward. And what this does is it increases the surface area of the cells, and by doing so favors the diffusion. Now, of course, these cells, these erythrocytes, are highly important for carrying oxygen as well as carbon dioxide. So a large surface area favoring diffusion makes them work that much more effectively. The membrane is quite flexible. This is important because erythrocytes have to be able to bend as they move around through branch points in blood vessels, especially when they get into the capillary bed. They have no nucleus or organelles, no mitochondria. There is no anaerobic glycolysis going on in these cells. They're pretty much a bag of cytoplasm that also consists of hemoglobin, which we'll be talking about shortly, which of course is responsible for carrying oxygen and CO2. There are some enzymes in the erythrocytes, glycolytic enzymes, which are involved with the energy, meeting the energy needs of these cells, and carbonic anhydrase, which is involved in acid-base regulation. And of course, the all-important hemoglobin. Now, what we show here is the hemoglobin molecule, which of course exists within the erythrocytes. And each hemoglobin molecule has 4 subunits. So here's one, here's another, and these are sort of stacked on top of 2 more, which are somewhat hidden. This is part of one here and part of another that are somewhat buried by these first 2 that I drew in. Each of these 4 contains 1 molecule of heme. And this one is hidden back down in here. This shows a detailed structure of one of the heme molecules. Heme molecules, of course, contain iron, which allows them to have the oxygen binding capacity that they do to permit them to bind to oxygen, which of course is the main function of hemoglobin. So let's take a look at some clinical problems. Anemia is when the blood has a decreased oxygen carrying capacity. That is, when there is either fewer red blood cells or the blood cells contain smaller amounts of hemoglobin, therefore a diminished ability to carry oxygen. So anemia is reduced hemoglobin, and it can be due to several different types of problems: dietary problems, loss of blood or hemorrhagic anemia, damage to blood cells, or we call hemolytic anemia, or a failure to produce mature blood cells, which we call aplastic anemia, or kidney disease, what we call renal anemia. So we'll go through each of these. Dietary anemia can be iron deficiency, when there's not enough iron being consumed, and of course the all-important iron part of the heme group. If you have less iron, you have fewer heme groups forming. That'll decrease the amount of hemoglobin. And then vitamin B12. Vitamin B12 is involved in absorbing iron from the diet. And what's called pernicious anemia is when there is a reduction or lack of vitamin B12, so that a person cannot absorb iron even if they have an iron-rich diet. Hemorrhagic anemia is due to hemorrhaging, that is bleeding, blood loss. Hemolytic anemia, a couple of examples, are malaria or sickle cell anemia. These are problems where in both cases we have malaria. We actually have a parasite that destroys red blood cells, or in sickle cell anemia, the hemoglobin actually takes on an unusual shape that gives it a sort of sickle or crescent shape that pierces the membrane of the red blood cell and therefore causes, of course, damage to the red blood cell. In both cases, we have lysis of red blood cells, which is why we have the name hemolytic anemia. Aplastic anemia is due to bone marrow defect, where we have a failure to produce mature red blood cells. And part of that maturation process is the formation of the hemoglobin. Renal anemia is due to kidney disease. There is a substance, erythropoietin, which is a hormone. If you remember, the kidney is a secondary endocrine organ. And it is called that because it in fact releases this hormone called erythropoietin, which causes the production of erythrocytes, hence the name erythropoietin. And in kidney disease, especially, for example, patients that have had cancer chemotherapy treatment, which can cause some kidney damage, there is a diminished production of erythropoietin and therefore an anemia associated with that. Platelets are involved in hemostasis, that is the control of bleeding, especially from injured blood vessels. So if there is, and you should probably look along in your textbook at these charts because they don't show up well on these slides. But if you have damage to a blood vessel wall, one of the first things that happens is vascular spasm or vasoconstriction as shown in this slide. What happens is that the blood vessel constricts, and of course this actually helps to work very much the way a tourniquet does. It reduces blood flow through the blood vessel, which helps to diminish the amount of blood loss. This is an intrinsic vascular response, that the smooth muscle responds to the damage and causes this response, and it's also augmented by activation of the sympathetic nervous system. Of course, you recall that sympathetic nervous system causes constriction of blood vessels, which is going to help augment this vasoconstriction. Also, the endothelial layer of the blood vessel becomes sticky. And that's going to be important because platelets are going to adhere to the endothelium. And when they start doing that, the platelets are going to release paracrines, including adenosine diphosphate or ADP, serotonin, epinephrine, which of course itself is a vasoconstrictor, as well as other chemicals that are necessary for blood coagulation or clotting. In addition, the platelets are going to start aggregating, forming what's called a loose platelet plug. And ultimately, we're going to have a formation of a reinforced platelet plug, and this is what we call clotting, that a fibrin mesh is going to form that's going to strengthen the loose plug. So let's go through this in a little bit of detail here. Have damage to a blood vessel wall. Collagen, that is part of the blood vessel wall. So let's jump ahead in this diagram. So we have collagen here as part of our blood vessel wall that now is going to be exposed to the blood. That's going to cause the platelets to start adhering to the endothelium, as well as release of some of these factors we've talked about. The whole point here is to produce temporary hemostasis, that is, to produce some sort of a plug that is at least temporarily going to block this opening that we've created in the blood vessel wall during injury. In addition, there are what we call tissue factors. These are substances that are released by the tissue itself. Of course, the blood normally is going to be confined within the blood vessel. The only tissue it has contact with is the endothelium. But damaged endothelial cells or any of these other cells that are part of the blood vessel wall would not normally have contact with the blood. When blood does have contact with these, we refer to them as tissue factors, and they activate some of the coagulation cascade, which is going on in parallel to what's happening with the platelets and the vasospasm. And these various substances that are activated are ultimately going to produce the formation of thrombin. Thrombin is a protein that produces a thrombus. Now, if you think about the name, technical word or term for a blood clot is a thrombus. So a protein that produces a thrombus is called thrombin. And it's actually going to cause this fibrin mesh to be formed that's going to strengthen this loose plug that was forming with the platelets. That is, we have this loose platelet plug forming here. And then when fibrin moves in, it's going to produce this sort of meshwork. It's going to help reinforce this loose collection of platelets. And this is going to reinforce our platelet plug, and as well as starting cell growth and tissue repair, which is also going to start activating other factors which are going to cause this reinforced plug to ultimately dissolve. So fibrin, which was formed by the thrombin initially, is going to be slowly digested, and as the clot dissolves, and ultimately we're going to have a fully healed blood vessel. So let's take a look at this process in some detail. So the first step of coagulation is platelet plug formation. That is, the platelets are going to start adhering and filling up this hole. So we have blood vessel damage starting this off, which is exposing what we call the subendothelium. That is all of this tissue that is under the epithelium. It's, of course, the connective tissue, smooth muscle, etc. A clotting factor called von Willebrand factor is going to be released, and it will bind to collagen fibers. The platelets then will bind to the von Willebrand factor. So that's going to start this formation of this loose plug of platelets. Platelets are going to start adhering, going to become stickier, which is going to help them to bind to each other, and they're going to start releasing paracrines. Now, these are substances such as thromboxane A2 and adenosine diphosphate, as well as some of these others that we listed back here on the previous slide, all of which are going to work together to assure that this soft plug forms and ultimately move on to the next stage when we're going to start moving toward formation of a longer lasting plug. So we have our aggregated platelets that are releasing secretory products, ADP, which produces a positive feedback, which is why we have the plus mark here. It's going to increase the stickiness of the platelets. Serotonin is being released. It's going to cause vasoconstriction. So, of course, it's going to help cause the blood vessel to constrict, which is going to help reduce the amount of pressure being applied on the plug. Epinephrine is also going to enhance vasoconstriction, and then a variety of other chemicals that are going to help facilitate the process of blood coagulation. And then the thromboxane A2 is shown here, which again is also involved in positive feedback. So once we have the platelets starting to stick here, we have these two positive feedback factors, that is ADP and thromboxane, are going to increase their stickiness. So you're going to get more and more platelets sticking together to help fill up this hole, forming what we call a soft plug. And this is the second step, what we call aggregation. Ultimately, aggregation is going to lead to the formation of a permanent or fairly permanent plug, something that will last for quite a few days. Then ultimately, we're going to have to both dissolve the old plug as well as you have other areas nearby. Let's go back here. We have areas here on either side of the tissue damage where we don't want clotting to occur. We want clotting to occur only where there is a hole in the vessel. So there are factors that are involved in preventing the formation or the spread of the platelet plug. These are chemicals that are released by the endothelium, such as prostacyclin, nitric oxide, that are produced by healthy, non-damaged endothelial cells, and they help to prevent platelet coagulation. In addition, there are other substances that will be released at the site of the clot that will help to dissolve the clot as the surrounding tissue heals. So now we're going to be moving on to other blood cell types and their role in immune function. So the leukocytes, that is white blood cells, their main function is, of course, defense of the body and the immune system. They're going to defend against pathogens. They're going to identify and destroy cancer cells, which of course are abnormal human cells. And they're going to engage in what we call phagocytosis, that is destruction of both cells and debris from dead or injured cells. The types of leukocytes we've mentioned already, they are the neutrophils, the eosinophils, basophils, monocytes, and lymphocytes. Now this is one classification system. They can also be classified in another way, that is by cells that have granular appearance, which we refer to as granulocytes, and those that do not have a granular appearance, which we call agranular cytes. The granulocytes have granules or inclusions in the cytoplasm. These are basically very much like we talked about in neurons, vesicles, if you will, that contain various factors that are important for these cells' function. So neutrophils, eosinophils, and basophils are all classified as granulocytes. Monocytes and lymphocytes do not have granules, and therefore they are called agranulocytes. We'll be talking about the specific functions of each of these cell types in a few slides. the synthesis of leukocytes, we have stem cells, and these are not exactly the same types of stem cells you hear about so much in the newspaper. Those are stem cells that can virtually become any kind of tissue within the human body. What we have here is a hematopoietic stem cell. That is a stem cell that will become some sort of blood cell. But that's all. It cannot become a brain cell, it cannot become a stomach cell or a muscle cell. So we have our hematopoietic stem cell, which is either going to become a myeloid stem cell or a lymphoid stem cell. Lymphoid stem cells ultimately become lymphocytes. All other cell types we've talked about start out as a myeloid stem cell. So erythrocytes, red blood cells, macrophages, neutrophils, basophils, eosinophils, and platelets all originated from a myeloid stem cell. And there are various factors or paracrines, what we call colony-stimulating factors, that are released by these different cells that control the direction of the proliferation and differentiation. That control the proliferation and differentiation of these stem cells into specific cell lineages. T lymphocytes are lymphocytes that migrate to the thymus gland, and that's where they develop full maturity, and that's why they are called T lymphocytes, because their maturation occurs in the thymus. B lymphocytes, which are not mentioned on this slide, mature in the bone marrow. So the B for B lymphocytes refers to the fact that they mature in the bone. And as I said, the development of all of these different cell types, starting from the hematopoietic stem cell, all of these different possible lineages is controlled by what we call colony-stimulating factors. And the colony that we're referring to here is a large number of some specific cell type that is reproduced by these parent cells. So neutrophils represent the largest proportion of leukocytes, that is 60 to 80%. They act as phagocytes, that is, they help to engulf microorganisms, abnormal cells, foreign particles, and destroy them. That is, they digest whatever it is that they engulf. They also secrete cytokines, which act as paracrine signals that help to activate other cells within the immune system. Individual neutrophils have a lifespan of about 7 to 10 hours in which they're circulating in the blood. And then they'll migrate to tissues for a few days where they have other actions. And they increase their number during infections. Eosinophils are a very small proportion of leukocytes. They secrete enzymes that kill parasites, as well as contribute to tissue damage and allergic reactions. So, in addition, they also have some phagocytic action, but it's not their main mechanism of action. The main mechanism of action is secretion of these enzymes that kill parasites. And notice again, parasites, not bacteria, not viruses, but parasites. So they defend against parasitic invaders, such as worms. The granules contain toxic molecules, that is the enzymes we talked about up here, that attack parasites. Basophils, very small proportion of leukocytes are basophils. These are not phagocytic. They may defend against large parasites by releasing toxic substances. But largely they contribute to allergic reactions by the release of histamine or heparin, which are at least two of what we call the chemical mediators of inflammation. There are others that are also called inflammatory mediators, but these are the two main ones that are released by basophils. Monocytes only represent about 5% of white blood cells, and they are phagocytic. Newly formed monocytes circulate in the blood for just a few hours, and then they migrate to tissues where they become what we call macrophages. Macrophages can either wander around in the tissue or remain fixed in one place. And as the name suggests, macrophages, these are large phagocytic cells, which, just as we talked about with other phagocytic cells, engulf foreign cells, damaged cells, etc., as well as bacteria, and help to destroy them. Lymphocytes are a special subgroup of leukocytes that live within the lymphatic system. They represent a fairly large proportion of leukocytes, and they are 99% of the cells that you find in interstitial fluid. There are three major types: the B lymphocytes, which, as I said, mature in the bone marrow; T lymphocytes, which mature in the thymus gland; and then null cells. Plasma cells, which are a form of mature B cells, secrete antibodies. So that's another thing to keep in mind, that these release antibodies. And antibodies bind to antigen, which is bits and pieces of protein from foreign cells, and in so doing, mark them for destruction by T cells. Now, within T cells, there are several types we call helper T cells that themselves secrete cytokines that actually help other types of immune cells to do their job, primarily what we call the cytotoxic T cells that, as the name suggests, kill cells. So that's the function of the cytotoxic T cells. Cytotoxic T cells secrete factors, that is, chemicals that lead to the death of infected cells and tumor cells. Null cells are also called natural killer cells, secrete factors that lead to the death of infected cells as well as tumor cells. So there's a certain amount of overlap in what some of these different cell types do. And of course, one of the benefits of that overlap is that two different cell types working to a common purpose are more likely to achieve that final goal than just having one cell type by itself. B cells produce antibodies. And as we show here, we have some B cells that, when exposed to antigen, which again is particles of protein from either foreign cells or bacteria or the like, B cells, as shown here, have antibodies. These little Y-shaped structures are how we always portray antibodies. And they have binding sites. The end of each of these antibodies has binding sites, just as we talk about receptors for hormones or neurotransmitters have binding sites. These binding sites are specific for the specific molecular shape of individual types of antigen. And when the B cells detect the presence of these antigens, they increase in number and they differentiate into either plasma cells, which is also called effector cells, because they produce the effect of producing more antibodies, or memory B cells. And memory B cells are pretty much the same thing we started out with up here, that these have the antibody attached to them and therefore are maintaining the memory of what this antigen is that we want them to react to. So these memory B cells will circulate around until they again encounter antigen, in which case they repeat this process of proliferating, that is, increasing in number and differentiating into either plasma cells or a new batch of memory cells. Again, the antibodies, another name is immunoglobulins, which is abbreviated capital I with a lowercase g. And the function of antibodies is to mark invading cells, bacteria, whatever, for destruction. The T cells are the cells that are going to engage in the destruction. They are going to directly damage foreign cells as well as bacteria. They also, when they contact infected, mutant, or transplanted cells, they will kill them off. And they develop into what we call cytotoxic T cells, which are the ones that cause destruction of these cells, that destroy target cells. Now, the targeted cells, in some cases, are marked by the presence of antibody. This whole response, though, can take several days. There are also secretory products that can be produced by T cells that will form pores in target cells. And by forming pores in them, they actually cause lysis of the target cells. Null cells most are what we call natural killer cells. And that means that, like the T cells, they are cytotoxic, but they seem not to require the cooperation of B cells or other cells in marking the target cells. Null cells are primarily functioning in defending us against viral infections. And when they attack virus-infected cells, so they're not killing the virus, they're killing the cells that have the virus in them, and they cause lysis of the infected cell. But of course, this, in the long run, will destroy the virus itself. Null cells are fairly rapidly acting. They're part of the early immune response. Another type of immune cells, what we call mast cells. These exist primarily in the skin and mucosa, and they secrete histamine as well as other substances. But when you have an allergy to something, it is largely this histamine that's being released that's causing that itchy sensation that you experience. Histamine causes blood vessels to dilate. It causes the blood vessels to dilate and causes them to become leakier. It makes the blood vessels more permeable, in large part to allow some of the other immune cells to escape the bloodstream and to get into the tissue to help protect against whatever it is that initiated the immune response. And not shown in this diagram is dendritic cells. These are similar to macrophages, and they are therefore phagocytic. And their function is primarily to activate certain types of T cells. We don't fully understand what they do, but we do know that they help in the immune response by their activation of at least some of the types of T cells. Now, a lot of what I'm covering on this aspect of the immune system is not covered so much in the chapter on blood in your textbook, but it's covered on the chapter on the immune system, which we will not be discussing beyond this lecture. I'm just giving you a very broad sort of overview of immune system function, which is a very complicated system. We just don't have time to go into it in great detail, and therefore it would be very difficult to understand. So I'm giving you a broad overview with this, but I encourage you to at least read some of the early material in the chapter 24, I believe it is, on the immune system, if you would like to learn more about it. Some of the material that also is discussed in that chapter is what we talk about as specific versus non-specific defenses. The non-specific defenses are non-specific because they defend against all pathogens, not just specific pathogens. If you remember when we talked about proteins and protein binding, the idea of protein specificity, that certain proteins will only bind certain ligands. So the specific defense system has that same property. It will only bind certain ligands. It will only recognize certain antigens. It will only respond to certain types of foreign cells. Whereas the non-specific system protects us against everything. So the non-specific system generally occurs outside the lymphoid tissues, primarily in the skin and mucosa, where, of course, it places these systems at a great advantage for encountering material in the environment. Some of the responses produced are inflammation, the release of interferons, which are also involved in defense against viruses. Natural killer cells are part of the system as well as the complement system. The complement system is a very complex system that complements the function of T cells in the specific defense system, which is why it's called complement system, because it complements the specific system. And it largely causes lysis of bacteria by causing the production of pores through a system called the membrane attack complex. Which is also abbreviated as MAC. Specific defenses. This is what we mean by the immune responses that I've been discussing up until this point. They defend against specific pathogens, and this all occurs within the lymphoid tissue. The cells involved are the lymphocytes, the B lymphocytes, which are also responsible for what we call humoral immunity. This is antibodies, called humoral immunity because these are chemicals that are carried throughout the blood, which is technically what the word humoral means. And then T cells, which are involved in cell-mediated immunity, because it is the T cells themselves, especially the cytotoxic T cells, that kills off foreign cells, bacteria and the like. Whereas B cells release antibodies, which are chemicals, proteins, but they're not cells, that mark foreign cells as well as other types of pathogens for destruction by T cells.